This is the third recording on molecules of life. This one is about proteins. Proteins are essential for all living things. Proteins are some of the major building blocks of cells and parts of cells and organs. Okay, the monomers of proteins are called amino acids. They're called amino acids because they have an amino group on one end and a carboxyl group on the other end. They're both attached to a central carbon called the alpha carbon, and there's either a hydrogen or another functional group added here, and then each amino acid has a separate R group. There are 20 different amino acids. The R group is a side chain that's attached to that central carbon. Some amino acids have more than one functional group attached to the central carbon, but there are 20 different ones that have different R groups. R is just a variable, kind of like X is in algebra. The R groups that give its specific properties to the amino acid there are two basic groups. There are the hydrophobic ones and the hydrophilic ones. The hydrophobic ones have side chains like methyl groups that are nonpolar, and the hydrophilic ones have polar side chains. The hydrophilic amino acids are able to dissolve in water. The hydrophobic ones are less able to do so. And that just has to do with the characteristics that the protein has and has to do with the shape that the protein molecule eventually takes. Amino acids are joined together by a special bond called a peptide bond. Now they're still joined by dehydration synthesis and separated by hydrolysis, but the special bond, the peptide bond, occurs between the carboxyl group of one amino acid and the amino group of the next one. The general structure of a protein, the chain of amino acids, is called a polypeptide. Poly means many, and remember this is a polymer because small groups put together, and so these are polymers of amino acids joined together by peptide bonds, so we call them polypeptides. Here we see the dehydration reaction combining the carboxyl group of one amino acid with the amino group of the next, and you see that it removes this hydroxyl group and this hydrogen to produce water, and then we have our, right here between the carbon and the nitrogen is our peptide bond. Special kind of bond. Now there are lots and lots of different kinds of proteins here. It says tens of thousands, and tens of thousands could possibly be an underestimate. But the proteins in your body and in all living things perform lots of different kinds of functions in your body. There are enzymes, which are made of proteins. Enzymes are catalysts that speed up chemical reactions, really, really important to how things work in your cells. There are structural proteins that give, like your hair and nails, tendons and ligaments and things like that are made of structural proteins. There are lots of others as well. Contractile proteins are what help your muscles contract and relax because they contract, they get shorter, and they get longer again. There are defensive proteins like antibodies that work in your immune system that help protect you from disease. And then there are transport proteins that move things throughout the body. An example of a transport protein would be hemoglobin, which is in your red blood cells that help carry oxygen from your lungs to the rest of your body in the bloodstream. So lots of different jobs that proteins can do, all of which are very important to appropriate functioning of the organism. Enzymes here are called biological catalysts, and they speed up biochemical reactions. Okay, there's a thing about enzymes that each one only works on a particular kind of substrate. The substrate is the molecule the enzyme works on. You can use enzymes to put molecules together or to break them apart. When we talk about dehydration synthesis, that occurs because there are enzymes that do put those molecules together and provide that reaction. Hydrolysis the same way. Any kind of reactions where things are being built or put together or taken apart involves enzymes of one kind or another. Now the thing about proteins can be very, very large. They're oftentimes hundreds and thousands of amino acids long, and there are two main types of shapes of proteins. There are globular proteins and there are fibrous proteins. The globular ones are enzymes and most other proteins, and the fibrous ones are things like keratin, like it shows down here in the lower right hand corner. The reason they're shaped like this is because of the interaction between those R groups. Sometimes there are hydrogen bonds that form between some of them. Sometimes there are other kinds of polar kinds of interactions. when we have those non-polar side chains, oftentimes they need to be oriented toward the inside of the molecule to stay away from water, because remember, inside of a cell, everything is dissolved in water. And so the hydrophobic ones would want to migrate to the inside. All of those things affect the function. If you change some of these factors that can affect them, like heat, pH, salinity, and things like that, what you can do is change the protein too much, and it's called denaturing the protein. Denaturation is the process by which the proteins kind of unravel and lose their shape, and they can't work properly if they're not shaped properly. This is what happens when you cook an egg. When you break that egg open, the egg white is just clear and kind of viscous, and when you cook it, it turns white and more rubbery textured. That's because you've denatured the proteins that were there in the egg white. There are 4 levels of protein structure. Okay, proteins have 4 levels of structure. Most proteins that function in your body have at least 3. Some of them also have the 4th level. The first level of structure is called primary structure, and this is the sequence of the amino acids in the polypeptide chain. Now, this sequence is coded for in the DNA and the genes of your body, okay? And changing the sequence often causes some kind of change in the shape of the protein because of the interaction of those R groups. This is an example of a primary structure of a protein. Each one of these little beads on this string here, the abbreviations there stand for different amino acids like valine and alanine and lysine and so forth. And that's just the primary structure of the polypeptide. The secondary structure is what happens when you make that chain and it starts to coil or fold because of hydrogen bonding between the different parts of the molecule. There are 2 main types of secondary structure, the alpha helix and the beta pleated sheet. And these occur because of hydrogen bonding between the hydrogen and oxygen atoms on the amino and carboxyl groups of different amino acids, not the ones that are next to each other, but a little bit down the chain. So here we have an amino acid. You see the amino acids here, and you see where the peptide bonds are between the carbon with the carbonyl group and the nitrogen. And what you see is the hydrogen bonding that can occur between some of these non-adjacent R groups there, or between the carboxyls and the hydrogens on the amino group and the carboxyl group. And that forms a curve called a helix, a twisted shape called a helix. Sometimes there are other kinds of actions that can occur, and that causes this folding or this pleating that can occur in the beta pleated sheet. Both of those are involved in secondary structure of proteins. The third level of protein structure is the tertiary structure, and the tertiary structure, the third level, has to do with the overall 3D shape of the protein, and this is what determines how the protein works. This occurs from other kinds of interactions between the R groups of amino acids, not just between the carbonyl carboxyl group and the amino group, but between the R groups of the amino acids. They can twist, they can turn, they can cluster together. The hydrophobic groups are going to cluster toward the inside when the protein is dissolved in water. The hydrophilic groups will stay on the outside. Sometimes there are other kinds of polar attractions between different parts of the molecule, and it causes a big change in the structure. You can see here, here's the alpha helix, here's the beta pleated sheet going back and forth, and this particular one is folded and twisted back together to make this tertiary structure, the overall shape of which is kind of like this. This is called TransferEatin, which is that polypeptide or part of this overall molecule. And finally, some proteins have a quaternary structure, and this is when you've got 2 or more subunits that go to fit together to make a functional protein. Sometimes the subunits are identical to each other, sometimes they're different from each other. Some of them contain parts that are not made of protein. For instance, hemoglobin is made of 4 subunits, 2 alpha subunits and 2 beta subunits, each of which has a pocket that iron fits in to form the heme part. That's the part that picks up the oxygen when it's functioning in your blood cells. This shows a quaternary structure of TransferEatin, that one that we saw earlier, and here's hemoglobin. And you've got the alpha chains, the red and the purple chains here are the alpha chains, and the beta chains are the pink and the green. And each one of them has this little pocket here where they have the iron, which is the heme group, and that forms the hemoglobin molecule, of which there are thousands inside of a red blood cell. to pick up the oxygen in your cells. Lastly, the protein shape determines its function. That unique three-dimensional shape is what determines how the protein is going to function. If you have proteins that are incorrectly folded, that can cause all kinds of problems, including diseases like Alzheimer's and mad cow disease. Sometimes the misfolding can be due to the incorrect amino acid being put in the wrong place, or sometimes it has to do with unfavorable conditions like pH or temperature, things like that. Denaturation can definitely lead to improper functioning, and that's what happens sometimes in some kinds of illnesses, because the high fevers or whatever can cause denaturation. And that concludes the lesson about proteins.